then a lot of time we'll talk about selection, but other than selection, there's another parameter, the N, the population size, also contribute to the polymorphism level in the population. And a lot of time we observe extreme case by bottleneck. So when the population just after a bottleneck, then the frequency spectrum will be very different. You will have a lot of new mutations at a very low frequency, because even for the positive selected site, they don't have the time to evolve to the intermediate or high frequency. So that's the common feature we observe when the population just experience a bottleneck. So this is just a list of different estimates of theta. So this is like all different theta. So as I said, the theta is for and mu, so by neutral evolution. So if the population, most of the variation is due to neutral variation, then all the estimates should be unbiased to represent the polymorphism level in the population. But sometimes the population experienced selection or population shrinking, a bottleneck. So the estimate might not be the same from different type of calculation. And that's the one we want to utilize to show the population history or recent historical events to the population we are interested. This is another way to look at the site frequency spectrum. So the site frequency spectrum in a population, actually there's certain level of nucleotide they evolve at lower, at sort of like lower, intermediate, and high frequency at all time. But the selection and both negative and positive selection push the frequency spectrum to like two different directions. So this is the results sometimes we observe in the population. So it's like a shaping by all the population parameters and the selection events. Based on this idea, Fei and Wu is interested in derive a different test statistic, look at more extreme to high frequency or at least intermediate to high frequency alleles. So this is the data from their analysis. So the black one is the observed number, the data they observed in the population. And you can see there's extreme case that have excess of lower frequency mutations and also some like high frequency mutations showing in the black bar in this slide. And the white one is the neutral expectation. And that was the idea leading them to test or just like using the frequency spectrum to test whether these genes or some of the genes are under positive selection. So the statistics test they did is using the pi and compare that to the low frequency. So the ones contribute to lower frequency theta. So the statistic they called H test. So because they use the pi is the heterozygosity. So they come from the H, using the H as an indicator to look at the polymorphism level. So that's another test. And you also could use a different ways to test the same thing and use the lower frequency theta to compare with the Waterson's neutral expectation, and they call the E test. So with all these tests, scientists or most people are interested in the genes show positive selection using the polymorphism data from a sample in the population. So we can do, so usually a lot of people will do variety of tests to look at. Then the paper I provide you in this lecture is the one to really discuss or at least list all the possible tests and tell you a little bit about how we can use those tests to detect the selection strings. And there are also sensitivity issues for each test. So for example, we all think, most of the time we think the D statistic, the Tajima's D statistic is not a very sensitive test. But if some other test, maybe it's too sensitive to any of type of the changes. So a lot of times in practically, people use a variety of tests or a combination of the tests to indicate or to try to detect the signature of positive selection.